Ma'am, little.
Okay, we did 5 kingdom classification, children, yesterday. You remember the kingdoms?
Yes, ma'am.
Yes,
Yes,
ma'am.
Name
ma'am.
them. Name them, beta.
Monera,
Monera,
Protista,
Protesta,
Animalia,
Fungi,
Plantae,
Plantae, and Animalia.
Fungi.
Good that you people started with Monera. Why did we start with Monera only? They are the highest in
Simplest.
Simplest one.
So we started with Monera yesterday. If you remember, I said keep ignoring the primitive ones, keep moving to the advanced ones. This is how we go ahead when we talk about biological classification. So we'll just study a little more about Moneras and bacterias. You know about them a little bit. Tell me what all you know about bacteria before I start with the class. Anything that you know about bacteria? Must have read a bit about bacteria. Yesterday we discussed about 2 kinds of bacteria. Okay, let's talk about them only. Tell me what were the 2 kinds of bacteria I discussed yesterday?
Archaebacteria, new
Archaebacteria, new
bacteria.
bacteria.
What was the difference?
The market bacteria are
Archaebacteria are
the
old
old
bacteria
Archaebacteria.
Archaebacteria are the old bacteria,
are
while
comparatively
new
newer
bacteria
bacteria.
are
and
the
new
newer
bacteria
one.
are
The
derived
primitive
from
ones
them.
are the archaebacteria and the
Archaebacteria.
modern ones are the new bacteria. Have you heard about or come across words like gram positive and gram negative bacteria?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
You must have read about them somewhere. Hmm?
Yes, ma'am.
Did you try to find out what
Yes,
is the
ma'am.
difference between gram positive and gram negative bacteria?
Yes, ma'am.
Anyway,
The one which take the stain are known as gram positive, and which do not take the stains are known as gram negative.
okay, very good. Those who take the gram stain, they are known as gram positive, and those who do not take the gram stain are known as gram negative. Today we'll also be discussing a little later as to why some of them are gram positive and the others are gram negative. Good. Your previous knowledge till here is absolutely perfect. You know we have 2 kinds of bacteria. Those who will take up gram stain, they are known as gram positive. स्कूल से फोन कर रहे हैं बायो के पेपर को जरूरी बात होगी. Okay, beta. Tell me now. We are going to study more about gram positive and gram negative bacteria. You only told me. Does anyone know why some of them are gram positive, why others are gram negative? Hanji, beta. Anyone knows?
Ma'am, the stain which is there, crystal violet stain, the ones who take them have single, those are the single layered one which are gram positive, and which do not take them are the bilayered one.
Okay.
The stain was
Excellent.
given by H.C. Graham. matlab he differentiated the bacteria.
Very good. Very good. Excellent. She's given the perfect answer. We'll just be going into a little more details about what she's telling you about an extra membrane. That part thing we'll be going into details. Very good, beta. Too good. Chale, let's start. We're going to study about Monera. We know that all prokaryotic organisms we had put in the category of Monera. Some important features of Monera. Number 1, without any proper nucleus. Okay? They have, do they have genetic material or they don't have genetic material also? Tell me, do bacteria have
They
genetic
have
material or
genetic
they don't have?
material,
Ma'am, they have.
They
but it
have
lies
genetic.
free.
Yes, it is naked. It is not enclosed in a nuclear membrane. So
Membrane.
there will be no confusion over here that they do not have a genetic material. They do have genetic material. But because they do not have a nuclear membrane, they do not have a proper nucleus. So it is naked. It's just lying or floating in the cytoplasm. If we talk about this kingdom, Monera, we are talking about prokaryotes. The word prokaryote comes from two words, pro and karyo. Pro means primitive and karyo means nucleus. Why do we call them as a primitive nucleus? Tell me, why is it?
Because they lack nuclear membrane.
Because they do
Exactly.
not have a nuclear membrane and the material lies free.
Excellent, all of you. It is basically not organized nucleus. There is no nuclear membrane, although all the material is there, but membrane is not there. So it's a primitive nucleus. So this is the first one, that without a proper nucleus, that is why we call them as prokaryote. Coming to the second point, beta. They do not have any membrane bound organelles or structures like endoplasmic reticulum, mitochondria. They do not have these organelles, which are membrane bound structures. But all the material which is present in ER or mito, everything is there. The only difference is it is not present in proper membrane bound structures, but it is suspended in the cytoplasm. बेटा सब कुछ होता है, genetic material भी होता है, all the material in ER is also there, mitochondria material is also there. The only difference is कि membranes नहीं है. We do not have a nuclear membrane, so genetic material is floating in the cytoplasm. Similarly, ER material also, it is simply suspended in the cytoplasm. So we will say no proper membrane bound organelles are present. This is the third one. Let's move on to the fourth one. Beta, structurally it is very simple, but if you talk about functionally, it is very complex. Why do you think functionally they are complex? Think about a class where we have many students and some big task has to be done. Each work is assigned to one student or a group of students. The work becomes simple. Now think about you alone, given the work of the entire school exhibition. So complexity or simplicity, what is going to emerge in such a case?
Complexity.
Complexity.
So here, beta, what did we have? Basically, these are very small structures. There are no membrane-bound organelles. We know mitochondria has its own work. Endoplasmic reticulum has its own work. Here, we do not have proper organelles. Everything is mixed up. The entire material is suspended in the cytoplasm. So things are more complex functionally in such organisms. Although they are very simple structurally, because when we talk about a structure, you will simply have the outer membrane and you will have all the material inside, not even a proper nucleus. But functionally, they are quite complex. So which is the first point we need to remember about Monera? Tell me, beta. It's on your screens. You have to just read out.
They are without proper nucleus.
Very good. Break the word prokaryote. It comes from two words. Name the word. Give me the meaning.
Primitive nucleus.
Pro means primitive and karyon means nucleus.
Very good. Now tell me about the membrane-bound organelles in Monera.
There are no membrane bound organelles in Monera.
Okay. So where is all the material which is supposed to be present in the
cytoplasm.
nucleus?
It lies free in
In
cytoplasm.
In
protoplasm.
the cytoplasm.
Very good. It lies freely in the cytoplasm. Structurally, is it simple or complex?
Simple.
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 Simple. But functionally, it is
Complex.
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 functionally it is complex. So these are the things we have done till now. Let's proceed. कोई जल्दी नहीं है. आराम से समझ के चलते हैं. Let's talk about habitat beta. You know that the bacteria they are found everywhere. Cold habitat, hot waters, dry habitats, saline habitats, extreme salt, acidic, basic, everywhere, even in our body. Depending upon the survival in different conditions, bacteria are of two types. This I had already done with you yesterday. RK bacteria, that is the primitive ones, they can survive in extreme conditions, and eubacteria, which are the modern ones, they can survive only in normal conditions. If we take them to extreme conditions, they die. They cannot survive extreme cold, heat, salinity, acidic medium, or extreme basic medium. If the conditions are normal, suitable for them, they will survive the eubacteria. But Archaebacteria, they are the primitive ones, they can survive in extreme conditions. So on the basis of this survival in different habitats, bacteria are classified into how many categories?
2.
What are the two categories?
Archaebacteria and eubacteria.
Archaebacteria and
Archaebacteria and
eubacteria.
Archaebacteria and eubacteria.
Which ones can survive in adverse conditions, extreme conditions?
Archaebacteria.
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 And those which cannot survive if taken to extreme conditions, they die. Those bacteria
Eubacteria.
are known as, what are they called? beta?
Eubacteria.
Eubacteria. So till now, when we are talking about Monera, we have discussed about two types of bacteria. Can we have their names again?
Archaebacteria and
Archaebacteria
eubacteria.
and eubacteria.
Archaebacteria and eubacteria.
Bacteria and
Very good.
eubacteria.
Now I'm moving on to the third important member of the Monera family. Till now, everybody is confident. We are talking about Moneras, we are talking about bacteria. We have already done two main members, that is Archaebacteria and Eubacteria. Now come to the third bacteria, beta. This is Cyanobacteria. What is the other name? I'm sure everyone knows.
Blue-green algae.
Blue-green algae.
Yes. Why are they called blue-green algae? They are bacteria, na? Why are we calling them blue-green algae?
They are found in many colors.
Okay, very good. Others, please tell me, why have you ever thought why cyanobacteria are called blue-green algae? They are not algae, they are bacteria.
They can photosynthesize.
They can prepare their food.
They have
Okay, they can
different pigment.
photosynthesize, but then why are we calling them blue-green? This is correct. They can photosynthesize. You're absolutely right, beta. But
They
why
have
are we
different
calling
color
them
pigment.
blue-green? Why not orange, red, or purple, blue, black? So anything we could have given. Why blue-green? It's very simple. Just guess a little, you'll easily get the answer. You know about colored pigments?
Yes, ma'am.
Yes.
Yes, ma'am.
Xanthophylls, carotenoids, you've done about colored pigments. So these bacteria, they contain colored pigments. And which are the two main colored pigments present? Blue and?
Green.
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 Yes, simple. They contain colored pigments, blue and green. And because of the presence of that chlorophyll, that is a green pigment, in abundance, they can photosynthesize. So cyanobacteria, blue-green algae, also included in Monera. They're photoautotrophs. They already have the green pigment chlorophyll. They use the solar energy and they can perform photosynthesis. So now we are done with three members, three important members of Monera. Come on, name all three of them, beta.
Archaebacteria, eubacteria, and cyanobacteria.
Beta, you speak out. That is the only way to learn classification. I'm telling you, if you sit to learn, you really find it very boring. This is entirely rote learning. So try that you speak out so that here only you have learned. So we are done with three categories. Name the two, what two color pigments will you find in cyanobacteria?
Blue and
Blue and
green.
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 Blue and green.
They are also known as photoautotrophs because they already have the green pigment and because of solar energy, they can perform. Now we are coming to the fourth most important member of Monera family. We generally have four categories only. This is the last one. That is Mycoplasma. Name the fourth member.
Mycoplasma.
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 It is also called PPLO. PPLO, pleuropneumonia-like organisms. What is the full form of PPLO?
Pneumonia-like organism.
They are very special. PPLOs are very special because they are the smallest cells without cell wall. They are the smallest cells. If we talk about cells without cell walls, then they are the smallest ones. PPLO, smallest cells without cell walls. So we have done the four main members of the first kingdom, Monera. Very quickly, let's name all four of them.
Archaebacteria,
eubacteria,
eubacteria,
cyanobacteria,
cyanobacteria, and mycoplasma or
and
PPLO.
And mycoplasma.
तो फर्स्ट टू मेंबर्स के बारे में हमें बहुत अच्छे से याद है आर्के एंड यूबैक्टीरिया बिकॉज हमने कल भी पढ़ा था सो वी आर नॉट गोइंग टू डू इट अगेन बट आई वांट यू टू टेल मी अबाउट द थर्ड मेंबर व्हिच वाज द थर्ड मेंबर
Cyanobacteria.
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 also called as
Blue-green
Blue-green
algae.
algae.
very good and what is special about this bacteria
It contains
contains
blue and green
blue-
pigments.
Blue and green
pigments.
pigment.
and it is able to prepare its own food, it can perform photosynthesis. Then we talked about the fourth member of Monera. What was the name?
Mycoplasma.
Mycoplasma or PPLO.
Mycoplasma.
PPLO. What is the full form of PPLO?
Pleuropneumonia-like
Pneumonia-like
organism.
organism.
Yes, they are also very special because when we talk about those cells without cell wall, then they're the smallest one. Let's talk about the shape of bacteria. They could be circular, oval, rod-like, comma, spiral, so many. Let's do this. You've done in junior classes also. If you see the circular one, it's called cocci or coccus. Anything, cocci, coccus. Then this oval shaped is your bacilli. This comma shape is your vibrae. This spiral shape is spirillum. And the thread-like is your mycelial. Okay. Can I have the main shapes? Circular. Circular, what do you call them?
Coccus.
Cocci or coccus.
Cocci.
Okay, the circular one is cocci or coccus. Then oval ones are
Bacilli.
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 comma types. What are they called?
Vibrio.
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 If you see this, this is spiral. What can we call them?
Spirillum.
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 And the last one is a single very thin thread-like structure. What do you call them as?
Mycelial.
Mycelial.
Yes. The majority of the bacteria around you is which shape, beta? Majority is which out of these five?
Cocci.
Cocci.
The cocci. That means
Cocci.
the circular ones. That means the circular. See, go back and see, this was your cocci, the circular one. Although we have all these shapes, but majority is this. So whenever we study, we mainly study about cocci only. Now, even these cocci or the bacilli or the vibrios, they are found in different arrangements. Okay, let me tell you this. Supposing these two are found together, these are which kind of bacteria? Which shape bacteria have I drawn?
Circular.
Circle.
Cocci.
What are they called?
Cocci.
Cocci.
Cocci or coccus.
Yes, if both of them are together, then we call them as Diplococcus. What will you call them as?
Diplococcus.
Diplococcus.
Diplococcus. And if they form a chain, if you see, they are forming a chain-like structure, then you call them as Streptococcus. Chain-like structure is called what, beta?
Streptococcus.
Streptococcus.
Yes, it is Streptococcus. And then if you have a cluster, if you can see this is a cluster of all the cocci, you can call them as Staphylococcus. What are they called?
Staphylococcus.
See, these words will remain constant. Diplo, strepto, staphylo. They are going to remain the same. ये change होता चला जाएगा. Suppose in coccus की जगह हमारे पास bacillus वाले हैं. Okay, let's talk about these ones. If two oval-shaped bacteria will combine, then we are going to call them as what? Diplobacillus. What will we call them?
Diplobacillus.
Diplobacillus.
ओवल वाला एक चेन बना देते हैं, लंबा सा चेन बना दिया with oval bacteria, then we'll call them as
Streptobacillus.
very good. And if
Streptobacillus.
we have a cluster of bacillus, then we'll call them as
Staphylococcus.
Staphylobacillus.
bacillus.
Staphylococcus.
चलिए इसको hide करते हैं. Let's talk about spiral वाला. No, let me talk about this comma one. This is single, so this is vibrio. If I attach one more comma-like bacteria over here, two vibrios together, what will you call them?
Diplovibrio.
Vibrio.
Diplovibrio.
Diplovibrio.
Diplobacillus.
You can call them as diplovibrio. And if I make a long chain, इसके नीचे एक chain बना लेते हैं बेटा vibrio की. What will you call them?
Streptovibrio.
Vibrio.
Vibrio.
Very good. And अगर एक cluster में इनको जमा कर दिया ये सारा का सारा vibrios, then we are going to call them as.
Staphylobacillus.
Vibrio.
So whenever you come across these words, immediately it should strike you that which shape bacteria it is. अगर coccus लिखा है, that means we are talking about the circular one. अगर vibrio है, that means we are talking about the comma one. Spirillum लिखा है, then we are talking about this one. And these first part of these will tell you how many and in what shape. So if I say diplospirillum, I'm talking about which shape of bacteria?
Spiral.
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 And how many spirals, beta?
2.
Yes, very good. And if I say streptospirillum, then it means what?
Long
Chain of spiral.
Cluster of spiral.
Strepto. Strepto is a chain of spiralums. And if it is a cluster, what will I call them as?
Staphylos.
Staphylo.
Staphylospiralum. This part is clear to everybody? I have just drawn one so that it's clear to you. Similarly, you can guess here the next parts also. Okay. Let us classify bacteria on the basis of flagella. This we had classified bacteria on the basis of shape, beta. We have already started classification. Dekho, beta. First, we did the 4 main members. Then we have started with classification where we had these 5 shapes. Now we are going to classify bacteria on the basis of presence or absence of flagella. Okay. What is a flagella? Let me first ask some things to you. You've come across the word flagella, I'm sure.
tail-like structure.
Thread-like structure.
Okay, what is
Which
its main
helps in
function?
locomotion. Locomotion.
Helps in
Locomotion.
Excellent. There is one more structure which generally helps in locomotion. What is
Cilia.
that?
Cilia.
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 Very good. What is the difference between cilia and flagella?
Cilia
सब
is
line
smaller.
पे पढ़ा होगा.
Cilia don't have.
Cilia don't have.
Cilia presents in a larger quantity while flagella is single unit or
Okay. Very good. चलिए. Very good. Whatever. At least he's sharing. Very good, bacha. Others? Any difference you can think about cilia and flagellas?
Ma'am, cilia are more in number than flagella.
Beta, speak. What did you say?
Cilia are
Tell me,
more
tell me.
than flagella.
Okay, that is what this child was also trying to.
Cilia are small, flagella is long.
Flagella perform only unitary action.
Cilia are hair-like structure, whereas flagella is thread-like structure.
Okay, beta. We were talking about flagella. You have told me quite some things about flagella. Do you know about structure of flagella? How it looks like?
Thread.
Thread-like
Thread-like.
structure.
Thread-like structure. Okay. And cilia also looks like thread-like. या वो थोड़ा अलग होता है?
Hair-like structures.
They are hair-like structure.
Okay. So almost what I wanted from you, you have already given me the answers. So now we can proceed further. We can also classify bacteria on the basis of flagella. I don't think it is there in your book, but चलो जल्दी से कर लेते हैं. Those flagella. There are some flagella where bacteria are there are some bacteria where flagella are present. They are known as trichus. What are they called?
Tricus.
Tricus.
And beta, what about the next one? Those which do not have
Atricus.
Tricus.
atrichus. Like you have cellular, acellular. Normally something lacking, we use the word a. So trichus and atrichus. These are some new words for you. I've tried to draw. देखो बेटा, trichus ones are again categorized into different shapes. अगर एक ही flagella है, one flagella, we call it as monotrichus. If we have two flagella, one at each end, एक इधर है, एक इधर है, then this is called as amphitrichus. What is it called?
Amphitrichus.
Amphitrichus. We have many flagella at one end only. We call them as cephalotrichus. And if we have many on both the ends, then we call them as lophotrichus. And pura body mein, all around if we have flagellas, it is known as peritrichus. So on the basis of whether flagella is present and where is it present, we have 5 categories. चलिए जल्दी से revise करते हैं. Again, you can check out with your books. If it is not there, those who don't want to do, don't do. Whenever I take an assessment, I take it only from the books. Chaliye beta, do it with me. If only one flagella is present, what are we going to call it as?
Monotrichous.
Monotrichous.
Now here we have two, but one at each end. एक इधर होना चाहिए, एक इधर होना चाहिए. What is this called?
Amphitrachus.
Amphitrachus.
Very good.
Amphitrachus.
Good children, we have many flagellas, but at one end only. इधर बिल्कुल नहीं है, इधर खाली है, कुछ नहीं है. Here we have lot of flagellas. Then this shape is known as
Cephalotrichus.
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 and we had a bunch here, we have a bunch here also now. This is Lophotrichus. What is this?
Lophotrichus.
Lophotrichus.
And he last wala hai, pura body ke upar all over we have these flagellas. Then this kind of an arrangement is known as
Peritrichus.
Peritrichus. Clear everybody?
Peritrichus.
Okay. Should I move further? If you are all okay? Okay. Let us talk about the flagella now. How does a flagella look like? What is the shape of a flagella? अंदर से क्या होता है? कैसा होता है? Beta, the flagella has three parts: basal body, hook, and filament. Name the three parts of a flagella.
Basal body,
hook, and
basal
filament.
body,
hook, and filament.
Filament.
Very good. Now, this basal, supposing this is the bacterial wall, ये जो दो line हमने बनाया है, this is the wall of the bacteria, you can say, or the membrane of the bacteria. Basal body अंदर होती है. This is embedded. This is actually embedded and it has rings. So what are the two things we need to remember about basal body? अभी और बना के दिखाया है आगे. Basal body is embedded inside the membrane of bacteria. बाहर नहीं होता है. ये वाला part जो है flagella का, ये अंदर होता है, inside the bacteria. So this is what we call as the basal body. This is embedded in the bacterial body inside the membrane, and it has rings also. What does it have? It has rings. 4 gram positive rings. We'll come to this. चलिए gram positive, gram negative बाद में करते हैं. पहले ये देखो बेटा. See, these are your membranes. What did I tell you? What these we had drawn? These are the membrane of bacteria. ये अंदर का पूरा part, inside the membrane, पूरा का पूरा. This is what we call as a basal body. If you see, this basal body has rings. What are these structures called as? What are these structures called as? Rings. Normally you have 6 rings. एक के ऊपर एक के ऊपर एक. You've got 4 plus 2, 6 rings over here. ठीक है बेटा? अब यहाँ पे आपका membrane खत्म हो गया बैक्टीरिया का. यहाँ पे ये जो structure start हो रहा है, this is what we call as a hook. What is this called as?
Hook.
Hook.
बैक्टीरिया की बॉडी के अंदर जो भी पार्ट है फ्लैजिला का, दैट इज योर बेसल बॉडी. जो कनेक्टिंग पार्ट है मेम्ब्रेन से अटैच्ड, दैट इज व्हाट वी कॉल एज हुक. एंड हुक के साथ दिस फिलामेंट, दिस थ्रेड लाइक स्ट्रक्चर, पतला सा थ्रेड लाइक स्ट्रक्चर व्हिच इज अटैच्ड इज व्हाट वी कॉल एज द फिलामेंट. व्हेन आई स्टार्टेड आई टोल्ड यू वी हैव 3 मेन पार्ट्स ऑफ द बॉडी ऑफ अ फ्लैजिला. बेसल बॉडी, जस्ट ट्राई टू एक्सप्लेन कि बेसल बॉडी अंदर होता है. देन वी हैव द हुक and the third part is the filament. What are the 3 parts of the flagella?
Basal body.
Basal body, hook, and filament.
Okay. See, if I take this as the membrane of bacteria, this and this, okay? Then this is the basal body जो अंदर है, जो attachment part है, this is your hook. And ये जो साथ में लगा हुआ है, this is what we call as filament. Now this filament is very special. बहुत लंबा होता है, बहुत लंबा, 100 times more than the cell sometimes. बहुत बड़ा होता है. It is made up of globular protein which is known as flagellin. This filament is made up of globular proteins. Globular proteins, and which globular protein is this made up of?
Flagellin.
Flagellin.
So ये अगर मैं अंदर से draw करके आपको दिखाया है, this is the filament which I have tried to draw. See बेटा, this is your filament. अंदर ये proteins हैं. Globular proteins which are arranged in the shape of a chain. And which is the protein I am talking about when I am talking about the filament? Which globular protein होगा बेटा ये chain में arranged?
Flagellin.
Okay. Flagellum can be 100 times longer than flagella. No doubt, बड़े बड़े तो होते ही हैं, but they can be very, very long also. Before I move on to gram positive and gram negative bacteria, very quickly the 2 topics which I have done. Bacteria can be classified on the basis or absence or presence of flagella. If flagella are present, they are called trichous. And if they are absent, they are called atrichous. अच्छा ये बताइए, if one flagella is present, we are going to call it as?
Monotrichous.
Monotrichous.
एक इधर है, एक इधर है. To each one end. We are going to call it.
Amphitrachus.
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 चलिए ये वाला देखो बेटा. Many at one end.
Cephalotrichus.
Good. What about these? दोनों ends पे group है.
Lophotrichus.
Lophotrichus.
All around we have flagellas. Then
Peritrichus.
Peritrichus.
three parts of the flagella. Very quickly. Name the three parts of the body of a flagella.
Basal body hook and filament.
And filament.
The part which is embedded inside the body of the bacterium is called.
Basal body.
Basal body.
तो ये वाला basal body हो जाएगा. And it has rings. ये अंदर दिख रहा है. This part ये जो attach करता है दोनों को. Filament and basal body. What is this called as?
Hook.
Then this is your filament. If I try and open and draw the filament, it is actually made up of globular proteins which are arranged in a chain. Globular protein chain. And which globular protein is this?
Flagellin.
Flagellin.
We have two kinds of bacteria, gram staining bacteria, which your friends have already given a lot of information, gram positive and gram negative bacteria. This stain was developed by Hans Christian Gram. We know the name of the person, Hans Christian Gram. Some bacteria beta इससे stain हो जाते हैं, then those bacteria are called gram positive. If they are not stained, we call them as gram negative. Let us understand कि ऐसा क्यों होता है कि कुछ stain हो जाते हैं, कुछ नहीं हो जाते. One of your friends just told you the main differences. उसको थोड़ा सा डिटेल में देखते हैं. See, this is a gram positive bacteria. Okay, see this gram positive bacteria membrane. This is the membrane that I've drawn. The cell wall of this bacteria is made up of peptidoglycin, which is very rich in an acid which is known as teichoic acid. ये वाला बेटा, ये जो इसका cell wall है, this is made up of peptidoglycin. Name the substance beta. Wall made up of.
Peptidoglycin.
And it is very rich in an acid, teichoic acid. चलिए इधर चलते हैं. ये वाला part again same है. Cell wall is again made up of peptidoglycin but without teichoic acid. Got the main first difference between both of them? This contains peptidoglycin is of course there, but it also contains, it's very rich in teichoic acid. Teichoic, teichoic acid. But idhar wala jo hai wall beta, cell wall, this is also made up of peptidoglycin, but without the acid. So this is one main difference between gram positive, gram negative bacteria structurally. The second difference which your friend had already told you. In gram negative bacteria, we have another layer. ये देखो बेटा, ये जो छोटा छोटा एक extra draw करने का कोशिश किया है, दो very untidy. Okay, this is another additional layer. This is made up of lipoproteins, polysaccharides. This एक additional layer आ गया negative में. This is made up of what? Lipoproteins and polysaccharides. चलिए, let us do the differences between both of them. Both the walls, when I talk about the cell wall of both gram positive and gram negative, both of them are made up of
Peptidoglycin.
peptidoglycin. The first one, that is gram positive, peptidoglycin is also rich in teichoic acid. Name the acid.
Teichoic acid. Teichoic acid.
Good. Teichoic acid is not present in the gram negative bacteria. Although peptidoglycin is there in the wall, but this acid is not present. Second and the most important difference, इसमें सिर्फ एक ये वाला wall है बेटा, peptidoglycin plus teichoic acid का. इसमें we have an additional membrane over here, which is made up of which two things I told you basically?
Saccharides and
This one, lipoproteins and
polysaccharide.
very good. बेटा दो membranes हो गए ना. जो अंदर वाला membrane है, this is made up of peptidoglycin but without teichoic acid. And ये जो बाहर वाला एक additional है, this is made up of lipoproteins and polysaccharides. ये याद रख लीजिएगा because ये आगे जाके staining में ये काम आएगा. Outer one is made up of which two things?
Polysaccharides and lipoprotein. Polysaccharides and lipoproteins.
Good, good. So we have got two differences. The first difference is whether acid is present or it is absent. And second is we have an additional layer in the negative bacteria. Staining process बहुत simple है. You fix the bacteria on the slide by heating them. This is called heat fixing process. This process is called heat fixing. Normally वो fix नहीं होंगे bacteria. So we have to fix them by heating them. So we have fixed the bacteria on a slide. Then we'll add this crystal violet for about 30 seconds to 1 minute, maximum 1 minute. इससे ज़्यादा नहीं बेटा. And then you need to wash the slide. Then you put iodine. This is gram iodine, a little different one. Again, the time has to be 30 seconds to 1 minute. And now you will not wash it, beta. उसको थोड़ा सा tilt करो बस. Tilt करो so that iodine remove हो जाए. You will see under the microscope that all of them are purple. ऐसा नहीं है. अभी तक हम लोग कह रहे थे some of them will take stain, some of them will not take stain. You will see that all of them are purple. Now you will wash them with 90% alcohol, that is acetone. आप सबसे अच्छा चीज ले सकते हो, लैब में easily available रहता है. Some bacteria will remain the purple color. They will retain the purple color. We call them as gram positive. Others will lose when you wash them with alcohol. We are going to call them as gram negative. Okay? Actually, superficially तो बेटा आपको color थोड़ा ऊपर से दिखाई देगा तो थोड़े time के लिए तो purple दिखेगा. Okay? अब if we talk about these, ये जो gram negative हैं, you treat them with safranin. Those who have not taken the purple color, you treat them with safranin. They will give you red color. That means जो gram negative हैं, they are stained by safranin, but they are not stained by gram stain. So what is the conclusion? ऐसा क्यों हुआ? That is what we are going to study very soon. But before I move on to the conclusion कि ऐसा सब कुछ क्यों हो रहा है, let me do this process now. What is the first step that you need to do, beta?
Fix the bacteria
Fixing
on the
them
slide
by
heating.
heat.
Yes, heat fixing process. We are going to do which process? Heat fixing process. Come to the next one now. What is the second step that you'll do?
Adding crystal of
We need to
violet.
add crystal violet.
Yes, we will add crystal violet and then we'll wash the slide. Then we'll add a special iodine. What is that iodine called?
Gram iodine.
Gram iodine.
Gram
And
iodine.
are we supposed to wash it or we'll just tilt it?
Tilt.
Just tilting.
Okay. And then you will get two bacteria. Some of them retain the, and then again you wash it with alcohol, 90% alcohol, acetone. Some of them will remain purple. These will be known as which bacteria?
Gram
Gram
positive.
positive.
Because they have taken gram stain. What was the name of this person? Gram? अभी पढ़ा था?
Hans Christian Gram.
Hans Christian Gram.
Excellent. Hans Christian Gram. And some of them will not remain purple after washing with alcohol. They will lose their color. So then you can treat them with another stain. Which is the other stain we have used?
Safranin.
And you'll get what color?
Red.
Red.
So beta, gram positive are stained with gram stain and they give you purple color. Those which do not get stained by gram stain, we treat them with safranin and we get red color. The conclusion is that gram negative wale jo hai, they can be stained by safranin, but they cannot be stained by gram stain. Now, why is this happening? This is what we need to know. कि why is this happening? कि ये तो हो जाते हैं easily stained, ये नहीं हो पाते हैं. Can we guess out before I go and give the explanation? Your friend had given you a clue also.
And because gram positive has one layer or gram negative has 2 layers.
So obviously it would not be easy for the stain to penetrate. And we'll also study as to why this stain, safranin, is easily taken by which one? Which bacteria am I talking about? Gram positive or
Gram
gram negative?
negative.
Gram negative. So when I talked about the composition, I told you इसको अच्छे से कर लेना because staining में काम आएगा. This very quickly. Both of them had this inner cell wall made up of peptidoglycine with teichoic acid, without teichoic acid. And the extra membrane, this was made up of what? Very quickly tell me.
Lipoprotein and
Protein and
polysaccharide.
polysaccharide.
Lipoproteins and polysaccharides. अब कोई भी चीज को अंदर जाना है तो यहाँ तो बहुत इजीली चला जाएगा because